How does GeneXpert MTB RIF Molecular Test Work? Imagine a test that can tell you if someone has tuberculosis and whether the bacteria are resistant to a key drug in just about 2 hours. That's what the GeneXpert MTB RIF test does. It's a powerful tool in modern medicine that uses molecular technology to detect tuberculosis bacteria quickly and accurately. The process starts with collecting a sputum sample from the patient, which is the mucus they cough up from their lungs. This sample is mixed with a special reagent that helps break down the mucus and kills any bacteria present, making it safer to handle. Once prepared, the sample is placed into a small, disposable cartridge that contains all the chemicals needed for the test. This cartridge is then loaded into the GeneXpert machine. Inside the machine, the process begins automatically. The bacteria in the sample are captured and then broken open, releasing their DNA. This DNA is purified and concentrated so it can be analyzed. The machine then uses a technique called real-time polymerase chain reaction, or PCR, to amplify specific parts of the bacterial DNA. It targets two main things: the DNA sequences unique to Mycobacterium tuberculosis and the section of the RIF gene that, when mutated, indicates resistance to rifampin, an important antibiotic used to treat TB. Fluorescent probes, known as molecular beacons, are used to detect the amplified DNA in real time. These probes bind to the specific DNA sequences and emit light when they do. The machine measures this fluorescence to determine if the bacteria are present and whether they have mutations linked to drug resistance. The entire process is automated, reducing the chance of human error and biohazard exposure. Results are typically ready in about 90 minutes, much faster than traditional culture methods that can take weeks. If the test detects Mycobacterium tuberculosis DNA, it confirms a TB infection. If mutations in the RIF gene are found, it suggests the bacteria may be resistant to rifampin, guiding doctors to choose the most effective treatment. The accuracy of the GeneXpert test is high, especially for cases where the bacteria are abundant, but it can also detect TB in smear-negative samples, although with slightly lower sensitivity. It requires minimal training to operate and is a safe, efficient way to improve TB diagnosis and management. This rapid detection helps in starting the right treatment sooner and plays a vital role in controlling the spread of resistant TB strains. Always remember, while this test is a powerful tool, it should be used alongside clinical evaluation and other tests to ensure comprehensive patient care. If you or someone you know is being tested for TB, consult healthcare professionals for personalized advice and treatment options.